Hello guys, welcome to the class of botany. Today we will be dealing with one of the very important topic, that is your biological classification. Though agreed that yes, you have already done the small basic of biological classification in your 9th standard, but the adverse knowledge with respect to your biological classification, we will be dealing it today. So let's start with your topic. If you can see, I have mentioned two categories here, kingdom Plantae and kingdom Animalia, agreed, which we are going to fill it up later. But let's go back and dig into the history that how this concept came into picture. Half of this thing we have already covered in a topic that is your taxonomy, which you can go back and find it out in the YouTube channel, which will be very helpful for you in order to understand this topic. Okay, agreed guys, when we are talking about your biological classification initially was started by your Aristotle. Aristotle was the first person, we have already done in the taxonomy class. Aristotle was the first person who said that yes, organisms can be divided on the basis of presence of RBC or the absence of RBC. Again, Aristotle was the first person who identified organisms on the basis of absence of RBC and the presence of RBC. Okay. So now after that it was Carolus Linnaeus who came into picture. Carolus Linnaeus was the person who said that there are two structures or there are two classifications. That means there are only kingdom Plantae and kingdom Animalia. Kingdom Plantae and kingdom Animalia. Basis on the above theories given by Aristotle, Theophrastus. So now Carolus Linnaeus has already given kingdom Plantae and kingdom Animalia. Carolus Linnaeus actually divided, or you can say differentiated, kingdom Plantae or kingdom Animalia on the basis of these general features. Let's talk about it. It was a very vague feature that he observed. Cell wall, he said it's a plant, so it is present. Of course, animals, absent. In locomotion plants, absent. Cell wall, locomotion, these are the features which Carolus Linnaeus observed in order to identify kingdom Plantae and kingdom Animalia. Locomotion absent, locomotion present. Mode of nutrition. Mode of nutrition, when we talk about the mode of nutrition, we talked about photosynthesis or not doing photosynthesis, but Carolus Linnaeus was not able to identify. Carolus Linnaeus was not able to identify. But when he said mode of nutrition, he said that yes, there is a mode of nutrition, but for Plantae, it will be not, or it you can say slow, and for it says yes, mode of nutrition was there. Contractile vacuole, when we talk about contractile vacuole, he said it that contractile vacuole was not present, was present. Organisms. Apart from these, what were the major important criteria that Carolus introduced? What he did during his two kingdom classification, during Carolus two kingdom classification, what he did, he put protozoa here along with vertebrates and invertebrates. Vertebrates along with invertebrates. Whereas this, he put everything here. Everything means literally everything. He put here fungi, gymnosperms, angiosperms, thallophyta, bryophyta, everything. He has categorized everything under kingdom Plantae. For Animalia, what he chose that only protozoa, vertebrates and invertebrates were there in kingdom Animalia. So it was two kingdom classification which was given by Carolus Linnaeus on the basic features, cell wall, mode of nutrition, locomotion, contractile vacuole, and the most important thing, what organisms he put under these criteria. Okay, so now let's move ahead. As we are talking about two kingdom classification, we need to know few more things which are very important when it comes to your competitive examination. The very first thing, the two kingdom classification given by Carolus Linnaeus was not able to identify, was not able to include into this was, first, he could not distinguish between photosynthetic and non-photosynthetic. He could not identify them. Could not. He could not identify. First thing is here, photosynthetic and non-photosynthetic. Second one, prokaryotes and eukaryotes. He was not able to identify, and yes, most importantly, unicellular and multicellular. These were the drawbacks in his two kingdom classification because he was not able to explain, could not explain. Okay, so Carolus Linnaeus, again, Carolus Linnaeus gave two kingdom classification. He observed certain features. On the basis of that, he gave certain criteria in order to identify them. But he was not able to explain these three features. That means photosynthetic, non-photosynthetic, prokaryotes and eukaryotes, and yes, most importantly, unicellulars and multicellulars. Okay. Now, let me give you a brief review or what kingdom given by whom. So let's move on to the next slide. I'll show you a slide here. If you look onto the slide carefully, agreed, characteristics of 5 kingdom of classification. When we say 5 kingdom of classification, everybody knows that it has been given by Robert H. Whittaker. Robert H. Whittaker, agreed. But when I'm talking about Robert H. Whittaker, he identified or he looked into these certain features. These were the features. But most importantly, before I start categorizing the features, we need to understand that who gave what. So let's do it. If you look at the Monera, I'll just rub this one off so that it becomes easy. Let's first talk about two kingdom classification. Two kingdom classification was given by your Carolus Linnaeus. Two kingdom classification was given by Carolus Linnaeus. Then we came to three kingdom classification, which included your Protista. So this two kingdom, third kingdom, it was given by Ernst Haeckel. It was given by Ernst Haeckel. Then we come to the fourth kingdom. Fourth kingdom means Animalia, Plantae, Protista and Monera. Fourth kingdom was given by Copeland. Fourth kingdom was given by Copeland. Now let's talk about your two kingdom, that is kingdom Plantae, Animalia, Protista, third kingdom, fourth kingdom including Monera, and now along with your fungi, which makes a total of 5 kingdom of classification, and this 5 kingdom of classification was given by Robert H. Whittaker. Now let's see what are the characteristic features that Robert H. Whittaker has taken. I'll just slide the board like this for you. Okay, if you can look into it. Let's first talk about the types that he has chosen. He has taken cell type, cell wall, nuclear membrane, body organization, mode of nutrition. So on the basis of this, now we are going to categorize different kingdoms. Okay. So let's talk about your Monera. Cell type will be, of course, Monera. Monerans are the one which majorly deals with your prokaryotes. Prokaryote, eukaryotic, of course eukaryotic. Then we have cell wall. We are talking about Monera. Okay, yes, exception to Moneras are always there, because now we are talking about your 5 kingdom of classification, but after this slide I will be taking care of your 6 kingdom classification as well, and the general basic criteria in order to distinguish the 6 kingdom classification from the 5th one. Now, we are talking about the cell wall, we are talking about Monera. Yes, Monera are non-cellulosic. Their cell wall or the membrane layer is made up of your polysaccharides and amino acid, sugar plus amino acid. Protista, cell wall might be present in some. Fungi, of course, chitin, non-cellulosic chitin. Plantae cellulose, and yes, in Animalia it is not present. Then we have nuclear membrane. Remember Monera, prokaryotes, bacteria family, so nucleoid is there, nuclear membrane is not there. Nuclear membrane absent, present. Then we have body organization. Of course, we being the Animalia, kingdom Animalia participants, so of course we will have tissue, organ system organization, but your Monera and Protista, they will have cellular level of organization, and the higher levels start from fungi. Last but not the least, the mode of nutrition. Prokaryote, autotrophic, but yes, some of them exceptions, chemoheterotrophic or heterotrophic can also be done. Protista, you have autotrophic, but some of them can also be heterotrophic. Fungi, solely heterotrophic. Plantae, of course, autotrophic, photosynthesis, and kingdom Animalia, that would be heterotrophic. So this is the basic level of classification which Robert H. Whittaker took in order to classify all the 5 kingdom of classification. Okay, so it was given by Robert H. Whittaker. So now moving ahead, what we will be dealing with is the introduction of 6 kingdom of classification. So when we are talking about your 6 kingdom of classification, I'm just going to write it down for you. 6 kingdom of classification. Now just see, how does it goes? We have Monera. I'm starting from 5 kingdom. Protista, fungi, Plantae, Animalia. Okay, let me make it easy for you. On the basis of its discoveries. Let's put Protista here. Then Monera. And then we have your fungi. This was your 5 kingdom of classification. This Monera was later subdivided into Archaebacteria and Eubacteria. When I am talking about Archaebacteria are the one which are also known as your primitive bacteria. Eubacteria, as the name suggests, Eubacteria are actually known as your true bacteria. How they distinguish between Archaebacteria and Eubacteria is on the basis of their cell wall, cell membrane organization. So the cell wall of Archaebacteria, of course, it is made up of polysaccharide amino acids, but it does not have one component, which you guys should know about this. Archaebacteria does not have peptidoglycan layer. Archaebacteria does not have peptidoglycan. Peptidoglycan is present in Eubacteria. Peptidoglycan is present in Eubacteria. And this new sixth kingdom of classification, 1, 2, 3, 4, 5, 6. This new kingdom of classification, sixth kingdom of classification, was given by Carl Woese. Was given by Carl Woese. Carl Woese was the same person who said that there is something which is known as domain of life. He said that there is a domain of life, and this domain of life included bacteria. It is Carl Woese who said it. Carl Woese was the person who said that there is a simple structure which is a domain of life, and this domain of life has originated in the form of bacteria. Archae means Archaebacteria. Eukarya means eukaryotes. Archae means Archaebacteria, Eukarya means eukaryotes, and normal bacteria. And this, he said that all of them had a common ancestor. And the common ancestor is known as progenote. Again, domain of life was given by Carl Woese. What he said that yes, there is a life form, a domain of life, which has a common ancestor that is known as progenote, and this progenote actually is subdivided into your bacteria, Archaebacteria, and then we have Eukarya means eukaryotes. So this is the all information with regard to biological classification. In the next simultaneous coming videos, we will be uploading lectures with regard to your protista, fungi, virus, and yet most importantly its life cycle along with all the 5 kingdom, and yes, the 6th one also. So hopefully this video has been helpful for you in order to understand the basic knowledge of biological classification. Thank you so much for watching.